Right? You know xylem and phloem, right? Xylem is there for water transport. Phloem is there for sugar transport, right? Sugar and other nutrients as well. So, yeah, remember this abscisic acid actually blocking these sieve holes on the sieve plane. The sieve tube, these holes are actually blocked by callous bush. Callous bush is something like that. This is callous bush. They go through the hole, and then the callus bush again. This is the cell wall. So the hole is there. Is the phloem transport not happening? They are just sealed off, right? So that is happening. We call this callous tissue. Like we call this this is actually happening under the influence of abscisic acid. Right? So it is a dormancy hormone. So then the plant is ready for dormancy during the winter. During the winter, they all seal and just not happening this. They're frozen, right? So that is abscisic acid is doing that. So even the potato, right? The potato growers know, right? The potato is filled with abscisic acid. When the winter comes, the aerial part of the potato plant, the aerial parts die. Underground tubers remaining, right? But the farmers take these tubers out, and if they actually put it back, just keep it in the back room or somewhere. If you put it back to the ground, they won't. They do not germinate, because still there are a lot of abscisic acid stored there. So abscisic acid make them for dormancy, right? So the tuber undergoing for dormancy, they are made for
so
that.
they don't do anything.
They don't do anything. So during the winter, right, winter temperature going so down. So during that, the low temperature only removing the abscisic acid, and the very deep part of the winter take the abscisic acid off, right? So then you need to put these tubers into fridge and put the temperature down, otherwise they won't germinate, right? So because that is doing by abscisic acid. So abscisic acid, by the way, right? So abscission is the falling of the leaf. So that is the senescence. Senescence is actually controlled by abscisic acid. So abscisic, the word meaning is falling off, right? Because that is how the very first time they recognize that is doing that, but then they recognize they're doing a lot of dormancy related things doing by abscisic acid.
Okay.
Right? And then last one is fifth one. It is a hormone, but it is a gas. It's a gaseous hormone, right? The gaseous hormone. Its name is ethylene. Ethylene, like polyethylene, you know. So the gas is that. So carbon, hydrogen. The petroleum product as well. Right? Yeah. So this ethylene gas is actually isolated from the plants undergo flooding. So this is a root system. The root system flooded, right? And you can see if the root system flooded and root system died. And the aerial parts die as well. If the root system is flooded for too long, what happened? These leaves become yellow and falling off and die. So during this time, the ethylene gas is producing in the root system, and through the intercellular spaces, it is come out to the aerial parts and they die.
Okay.
Right? So that is
Yeah.
the very first time they recognize ethylene such a way. So right. So
Okay.
they thought it's a kind of a destructive hormone. So there are constructive hormone as well as destructive hormone.
Destruct.
Yeah.
Okay.
But having said that,
Yeah.
it is not that simple as well, right? Maybe have you heard about plant tissue culture? Right.
I've heard of
Yeah.
it, but I don't exactly know what it is.
Yeah. Plant tissue culture is right. So you can use a small part of the plant. For example, in orchid, right? So
Yep.
you probably can get the orchid like a I just draw orchid plant and tiny little leaf here, and then
Yep.
what is under the tiny little leaf? That is a growing tissue, right?
Yeah.
These are the leaves, and this is a very tiny tender leaf, and beneath the tender leaf, you can get the shoot meristem, we call it. So meristem is word used for dividing tissue, growing part of the plant. So we call apical meristem, the root meristem and shoot meristem. Right? So they are the tissues which adding new cells into the plant. So if you dissect that, right, if you grow them in agar media, right, like that. So a small part of the meristem.
Yep.
Right. So you actually trial and error, you make these 5 hormones, and trial and error you can do it. Right.
Yep.
Actually, what you changing is the gene expression. Right. How do these hormones? How do these hormones working? They are actually stimulate and repress the expression of the genes. So they are pretty much signaling molecules in plants. All right. So that's why they put in here. So they are belongs to signaling molecules, obviously. So these small tissues you can actually changing hormones. You can develop it into kind of a balls of cells, cluster of cells like that. So they are consist of undifferentiated cells, right, in plants. So the undifferentiated cells, similar cells in animal body, we call them stem cells. So, so pretty much they are embryonic cells, right? Even human being at the embryonic stage, you actually the embryonic stage, you have only undifferentiated cells, right? So likewise. So then from these undifferentiated cells, you can cut it to any number, and you can make this callus. We call callus, right? CALLUS. Callus can make thousands of callus. So end of the day, you change your media.
They self-replicate?
Sorry?
Do they self-replicate?
They keep growing. They keep growing, self-replicating.
Oh, okay.
But then you can cut it off, and it can distribute to thousands of flowers, and then they grow the same way, right? Then you change the media, so they make new leaves, right? And then you change the media again, they make roots, right? And you can cut it off, cut it off, put it into new flowers. You can make thousands and millions of small plantlets. The thing is, they are all this clone of the same original plant. So this is what you call plant tissue culture. So the plant tissue culture is a very big industry, right? So if you can find the correct media, so you can replicate any number of plants. So orchid, for example, the orchid doing the tissue culture very successfully, right? So you get one particular beautiful flower, right? So then you can get a clone of that flower. So you can distribute all over the world the same color, same beautiful orchid plant, right? And also you can actually genetically modify those plants by inserting beautiful colored genes into the callus, right?
Yeah.
And you can germinate them and see what's the color of the flower. Or maybe you can actually, actually color of the flower is changed by inserting genes, so you know what color it's going to add. But then of course you have to see the flower to see the final appearance. Does it go to the places where you want them to go? Flower, its center is black dot, or the tip of the petal is drop there, so whatever it is. So then you have to look at the plant first, grow one plant, right? And if it is good, pick up the good one, and then tissue culture them. That's it.
Right.
So then you can get millions of plants. So that's how these days most of the things actually the plants are so marketed that way, because they do all tissue culture plants. So Germany, like European countries doing this, Japan doing this. So they sell their plantlets to very, very high prices, right? Carnation plants, roses, everything, right? So so the simpler the plant, the kind of orchid plant is a simple plant, tomato plant is a simple plant, like they are very easily being tissue cultured than perennials. What are the perennials? Like rubber tree, very hard. The Rubber Research Institute is trying to do that for many number of years, still can't. Because the complicated the plant, the complicated the changes, and complicated the trial and error thing. If you can read the genes, you can actually directly go to the changes. So, this gene require this gene require that, but we can't read the gene, that we don't have the knowledge is that much, not much developed yet. So that's why we do trial and error. So trial and error can do things like that, but developing a human being is not that easy. So because so many hormones are involved, even we don't know what they are. So plants are very simple living beings. So then 5 hormones, as I talked to you before. So combination of these 5 hormones, right? So different tissues, different species of the plants responding to these 5 hormones combination, totally different ways. So one hormone, abscisic acid and ethylene, kill those plants. But another hormone, another plants, like for example, bromelaceae plants, like pineapple plant. So ethylene gas cause them blooming, so usually very hard for bromelain, like bromelaceae plants, like for example, like pineapple family plants. They're not blooming easily, right? So the pineapple plantation, if you use carbide actually producing ethylene gas. So ethylene is a plant hormone, which is killing hormone, right? It's not a constructive hormone. But in this family, that hormone actually trigger all of the plantation to bloom. So then you get a lot of the fruit of the fruits, right? So I mean pineapple. So by ethylene gas. So that, like different plants responding to the same hormone different way. So that is why. So every single time the plant tissue culture, maybe you just Google them and see a lot of information. Very interesting. All right. So I'm going to stop the class now, right?